the deepest dive ever done in the Seychelles.
A record-breaking dive.
Beat it by nearly 1,000 meters.
Well, we've never done any dives in this trench, so this was a really groundbreaking, exciting dive for all of the Seychellois, especially on board.
All right, welcome to our very first episode of Deeper Dive, a show where we relive some of our most interesting and exciting ROV dives that we've conducted aboard our research vessel, Ocean Explorer, which you can see here in all her glory. So to kick things off, we've got a very cool dive for you, a record-breaking dive, in fact, from our recent mission to the Seychelles. And so as our ROV begins to descend here, let's have a quick round of intros. I can start. I'm Eben Hall, part of the OceanX media team. Andrew, how about you go?
I'm Andrew Craig. I'm the ROV program lead at OceanX, and I was conducting this dive with my team down to the Amaratis Trench for Sheena.
Yeah, and speaking of Sheena, go ahead.
Hi everyone, I'm Sheena. I'm from the Seychelles and I'm a deep sea biologist. I'm taking on a PhD in deep sea ecosystems, and this was a really exciting dive.
Nice. And Jarila?
Hi everyone, I'm Jarila Kamau Wang. I'm a research analyst for OceanX. I work in the science pillar. I mostly work shoreside, but I help plan expeditions and very excited to watch this dive.
And last but not least, Larissa.
Hi, I'm Larissa. I'm the eDNA postdoc here with OceanX. I work on the vessel targeting and dealing with all things molecular ecology. And I was part of the mission in the Seychelles where we did this dive and supported the scientists on board in their goals and achievements.
All right, excellent. And we are zooming straight down with this condensed version of this dive. So you can see the depth on the right-hand side of the screen there. So we're already down to 4,000, almost 4,400 meters in just a matter of a few seconds. Andrew, how long would it normally take to get down to these kind of depths?
Yeah, I mean, this is probably 2.5, 3 hours worth of diving so far. Very exciting at the start, very exciting at the end, not so exciting in the middle, unless something comes along. But yeah, you can see here, we usually go down about 35, 40 meters a minute. If we're doing repeated, very, very deep dives, we'll probably use a biodegradable sandbag so we can sort of speed up the process. But yeah, it's pretty long and painful to get down to these depths. But as I think Sheena will attest to, it's worth it when we do finally get there.
Yeah, Sheena, why are we diving in this place? What makes it special?
So the Amaratis Trench we've never done any dives in this trench. They've done a bit of mapping here back in 2020, and that's about as much as we know about it. So this was a really groundbreaking, exciting dive for all of the Seychellois, especially on board. So what we're not seeing as we descend is the large group of people that had accumulated into the ROV shack. Maybe to Andrew's dismay, because there was so many people there. And we were all, we were quiet in some places, chatty sometimes. And as we got further and further into the dive, it was just crazy because none of us could believe that we're actually diving in our own waters and it's being led by us and we're seeing these depths for the first time. So it was a very, very special moment. Even the boring parts.
Yes.
And how deep exactly are we talking? What was our sort of target depth?
Correct me if I'm wrong, Andrew. I think we were looking to go to about 4,000, so 4,500. We went a bit deeper than that. The deepest that we know of in the trench is about 5,300 meters. But we were playing around with time and operations, so we wanted to make sure that we got to see the bottom and some of the trench and be able to come up with enough time to be able to, go to our next spot.
Yeah, we have it's a 6,000 meter ROV, but yeah, exactly as Sheena said, we were kind of limited on time. It was the last day of ops, and we had to get back to port. So we had to we had to measure our time between record bottom depths and getting some time on the bottom, which is sort of like with these dives, is part of it, because 3 hours down, 3 hours back, a 12-hour shift doesn't leave you a huge amount of time on the bottom. So yeah, we have to make sure we can get as much done as possible.
And this was, I mentioned this was a record-breaking dive. So this was, what was the record, Andrew?
Yeah, prior to this, we were able to get the ROV to about 3,900 meters up in Svalbard, up in the Arctic Circle, into the basin there. Beat it by nearly 1,000 meters, so it's good, very good. And the vehicle held up. It was very impressive, no problems. We got straight down, which is pretty unusual as well with these super deep dives, to actually not have something fail or need to put oil into something, when you're going down to that sort of crushing depth. So yeah, very, very impressed with the dive, and was able to actually, set out a plan, and we actually were able to carry the whole thing out as we laid it out on paper, which is pretty unique as well.
Sheena, do you remember how you were feeling at this point in the dive, knowing that the bottom is getting close, the first dive in this special place?
So I was telling Maddie and the rest of the crew that, this has been one of my dreams ever since I realized that, hey, I can study the deep sea. And I was telling Maddie that, like, okay, now that we achieved this, I need a new dream. But what was really special actually was all of the whole team gathered. And as we were getting closer to the bottom, we started counting down to the bottom. So it was like, it was really, really epic, because it was like exploring space, so everyone in the room was so happy, and yeah, there were lots of shouts and screams and some crying. There was some crying in the room. I won't say who, but yeah, Andrew was definitely one of them.
It wasn't me.
He wasn't in.
Well, it looks like that altimeter is dropping down. It was at 75 meters before, and now we're sort of hovering around 40. Does that mean we're actually at the bottom or close to it, Andrew?
Yeah, I mean, like as it would sort of indicate, we're 40 meters from the bottom. So the altimeter comes in at about 65, 70 meters, depending on the bottom composition, and then it'll start counting you down in. So you got plenty of runway to know that you're getting closer and closer. And then the sonars on the front, which you can't see in this picture, but the sonars start painting a sound image of the seabed at the same time, and other things start to happen that you sort of know you're getting close, and people start to scream a bit louder and count a bit louder, and yeah, it's you're definitely getting close to the bottom. This is usually where things go wrong, and everybody is like this huge sort of and then we have to bring it all the way back up and try again. But this time, like I said, we made bottom, as they say.
Oh yeah, well, 15
It's
meters
crazy
to go.
how it's still so exciting. I mean, I was there for the dive, but now we had like the countdown on the altimeter, and I'm still like, oh my God, we're gonna see it soon, like right now.
The anticipation it's killing me, guys.
Should we start counting down?
Yeah, it's like 10 meters
It was
now.
quite funny when we were doing the countdown, because sometimes, you were going 10, 9, and it was taking longer than 10 seconds would take. So then we'd just be like 9 for a really long time. But yeah, it was, I think, that's something that we can all share. And Larissa was there and Andrew was there, and it's something that will stay with us. Yeah, for me especially, forever.
Oh, there's
Hey, here we go. Our first look at the bottom.
Mhm.
Very exciting. Okay, so like, what would be happening in mission control right now? We've reached the bottom. So what next?
Lots of hugging.
So after all the noise stops, now we kind of sort of settle the vehicle from an operational point of view. We kind of settle the vehicle, figure out on okay, what do we actually want to do now that we've made it down, and obviously we needed to get a couple of samples. We know and have a look around, and then kind of this is where Sheena takes over. Sort of like, okay, Sheena, we've made it down. Let's do some exploring while we have the time. You got 3 hours, go for it. So, and yeah, Sheena, I remember you taking over here.
Yeah, this was the most high pressure 3 hours of my life because you're just like, should we go straight this way, that way? So we just decided on doing a vertical transect as far up as we could get to. And yeah, at this point in mission control, everyone was very excited. There was lots of hugging going around, and some of us were writing things down. So what time we reached the bottom, what we're seeing when we reach the bottom. And yeah, the first thing we kind of landed on were these sea cucumbers, or gummy squirrels, which are really, really cool. And you're so used to seeing it on videos of the deep sea, that I was, in my mind, I was like, is this actual footage of the Amaranth Trench, or are we just watching some playback video from David Attenborough? So this was a really special moment, just landing and there being these sea cucumbers right underneath us.
Yeah, what is that part of it that's sort of sticking out of its head?
Yeah, it's like a sail, right? So it looks like, like sort of like a dorsal fin or something. It's just like this sail. And apparently the adults kind of use it time to time because they're mostly benthic, and they leave most of their life on the bottom. But when I've read up about them, it sounds like as larvae, when they're a lot younger, they will use it a lot more in their pelagic state than when they're adults. So it's really, really interesting.
I feel the name they have, like with gummy squirrel, that's just such a fit because that's exactly what they look like. And the sail just makes it look even more squirrely.
Yeah.
Gummy squirrel, of course, being the proper scientific name. Is that right?
Of course.
Of course, no. Psychropotes being the big group that they all belong to, but there's like different varieties of them, and there's still a lot that we don't know about this species. So yeah, they're actually really interesting sea cucumbers.
I see our lasers just kicked on. Andrew, what are those for?
So they're scaling lasers, so they're 10 centimeters apart. So anytime that we're looking at something from whether it's right beside it or up in the water column and we're looking down, we can put them on, and then we've got basically we can figure out size. So we know straight away that gummy squirrel is 10 centimeters apart across, so it's probably about 30 to 40 centimeters long. So yeah, just a very quick visual way of figuring out the size of something, because it's very difficult with the perspective from the cameras as to how big or how small things are, especially when you're zooming in on them and zooming out.
And Sheena, down here, do we know anything about their diet or if there are any predators down here on the gummy squirrels?
That's a great question. I mean, like most sea cucumbers, they're like detritivores, or they like feed in the sand. And I guess they have a very similar role to a bit like earthworms, where they rotate the sand around. It goes in them and through them, and so yeah, they're really important for these areas in these abyssal plains. Whether they're eaten, I have no idea. I don't know what eats them. I mean, I feel like they would taste not so great. I don't know. I mean, they look cute, but I don't think they taste as good as they seem.
They taste great, but the texture, oof.
I
Yeah.
bet, yeah, very squishy.
And it's interesting because you get like that purple variety, and then there was like a yellow variety as well. So, especially with the name Gummy Squirrel, I wonder if they would taste like gummy bears. That's what I imagine, at least
Yeah,
the different colors being
your
different.
grape and your lemon.
Yeah, maybe different flavors. Yeah.
Yeah, probably. But I feel like they would be like crunchy gummy bears, not like
Yeah?
the super soft ones. I feel they would have a little crunch.
What, because they're cucumbers?
I don't know. If you just look at them, like the texture is not like super smooth, so maybe they have like a little crunchy shell-ish situation.
Oh,
Oh,
okay.
okay.
What's happening, Andrew?
Let me see. Oh, I think we're going to do some scoop samples. Yeah,
Oh,
so we're going to pick up the our little scoop, and we're going to get some sediment samples for Larissa.
yeah.
Actually, that I didn't think about this before, but if you scooped the mud from behind the gummy squirrel, can you test that to see what's passed through it?
Yeah, I think that certainly is an option. Like if we would, scoop right behind where the gummy squirrels just were, I'd assume that they would leave behind quite a good amount of DNA in like probably having some like mucus layer outside, and then just shed that onto the sediment. So I'm assuming that we will have at least some content of gummy squirrel DNA in those sediments that we collected with the scoops. Yeah.
Quite cool. Yeah, so I mean, a part of our MO when we're doing any of these dives, whether it be in 4,600 meters or 40 meters, is collecting samples, and sort of this is very much dictated by the scientists who are on board at the time, and because of the facilities we have on board, like Larissa can speak more to it in a second, but we taking sediment samples or water samples or whatever and bringing them on board the boat is really important for us because we have the ability to be able to really analyze them and sort of really understand exactly what's in them. And I will pass you over to one of our experts because I am by no means an expert in this field. Larissa?
Yeah, that's actually yeah, probably the part where I tune in on the boat. So all the samples we collect with the RV or with the submarines, as you can see here with the sediment, which looks so beautiful. Like I just remember when it came back on board, I was just like, oh my God, that's so amazing. Deepest sediment I ever had. And it was so nicely colored and so fine. I actually kept a little bunch of it and made little keepsakes. But yeah, back to the science. So in that sediment that we collected here, we can find DNA traces from all the organisms that were in that habitat. So either on the seafloor or in the seafloor, and they leave behind their little DNA molecules by mucus layers or just by movement. They lose scaled or shed other sorts of secretions, just like humans would lose hair in a room. And we can collect those with the sediment and the water from those habitats, and then we bring it back on board where we can sequence the DNA that we find there. So we translate it into like a pretty much some sort of digital data that we then can compare with databases that allow us to tell what organism has shed the DNA in that environment, so we can infer what species do live where we sampled, which then kind of like bridges between the actual exploration of a habitat, but then like discovering what actually is in there, and we can run that all on board immediately after we collect those samples, which is super amazing.
Okay,
And I
very
think
cool.
that's
So this would
something.
be, yeah, go ahead,
Sorry,
Sheena.
Evan. I
No, go
think that
ahead.
was something that the government of Seychelles was really excited about, just the ability to grab a sample and be able to sequence it. And our fishery authority was really excited to find out what fish we were finding at the different depths. So Larissa was an amazing teacher here. She definitely trained up at least two of the scientists on board that are now in love with eDNA. In love.
That's amazing. I love to hear that. I love to pass on the word of the magical thing called environmental DNA because it's just the best.
I mean, for the kind of casual listener who sort of doesn't really understand the real implications of this, I mean, what it means is that we can go somewhere and not have to sit there and stake it out to sort of understand what's there. We can now go there and test the water and the sediment that is there to know what was there or what's in there, which is pretty major for our line of work, isn't it?
Yeah, exactly. It's probably the major benefit, especially in anything that like involves fieldwork or like probably many aspects of ecology and ecosystem assessment, where you with like traditional methods would rely on the species like being visibly there, so you can like observe them or record them um, on like devices or trap them, net them, so like capture methods, which are all based on the fact that the species are present in the very same moment you are present as well with the sampling technique and sampling gears. Whereas eDNA allows us to detect the species or taxon groups of interest, regardless of if we visually can observe them or if we can catch them with any sort of tools or gear. So the picture that you can paint in terms of biodiversity and community assessment, or for example fish stock assessment, is just a lot easier for us to do that because we do not rely on actually being there at the same time as the fish that we want to observe.
Okay, that makes a lot of sense. And I would assume many deep sea species might be sort of wary of the ROV and the lights and everything. So it seems like
Yeah,
having this
I would
ability
be
to.
shocked if suddenly out of nowhere a bright yellow light up robot drops into my backyard.
Okay, so it looks like our first sample was collected successfully, and then on with the transect.
Yeah, so at this point we're kind of like, you see the altimeter, we come up to about 2.5 meters, and then that sort of gives a really good overview of the bottom. What you're not seeing is some of the other cameras. We have some lasers looking down on another camera, so the transect is sort of in a sort of a pillar box of the bottom, a fixed camera, so that's always rolling, so that they can compare. Sheena can take the data away and compare, what the bottom looks like over a certain area with the lasers in it. So again, for habitat abundance and species counts and all of that, then that can be used even in these super deep depths. So it's all about comparing and contrasting, right, Sheena?
Yes, exactly. Thanks, Andrew, for bringing that up. Because what we're seeing in the water column might be very different to what we're seeing on the bentos or on the bottom. So it's really important that we can capture both of those things. And yeah, it will take a couple of hours, months, to go through all of the footage that we've collected to be able to paint a picture of what organisms we're seeing and why we're seeing them at those different depths. So some very exciting stuff coming on, but yeah, it will definitely take us some time to get there.
What do you estimate how many hours of footage do you have? Like right now, we're just looking at that one camera the RV has, but actually there are a lot more. So are you going to look at like all the footage from all the different angles, or are you going to like pick one of the cameras for a certain type of analysis?
That's a great question. We're gonna actually use only two cameras, so the forward-facing and the downward-facing cameras. So we'll look at the pelagic, so if there's any fish swimming around, and then we'll look at the benthic, so the corals at the bottom, or if there's any little critters hiding in the corals or in the sediment, and try and identify them as much as we can. It's really difficult sometimes, and then we'll paint a picture based on the community composition in the different areas and the different depths.
Cool.
So here's our other sampling method that we did collecting water at the bottom. This is a Niskin bottle, again using the manipulator, which are our hands underwater. As you can see, we're poking around. Our depth perception's a little bit tricky with camera feeds, so sort of putting the tiny little finger on top of a tiny little post is a lot of fun, but yeah, it can make you look a bit silly. So push down the plunger, the top and the bottom cap. There's a rubber band between the top and the bottom cap. So once you trigger it, that rubber band will go tight and trap the water in there. So now we've got a sample of water from 4,600 meters. Actually, I think we're going to get a second one. So 2 samples of water from 4,600 meters, which taken vertically, and then that will be transported all the way back, and then again that will be handed over to the science party, and then it's into the lab with that as well. Or what did we do with that? I can't remember what it was used for. Again, eDNA sampling, wasn't it, Larissa or Sheena?
Yeah, we did take those Niskins and filtered them over like a very, very, very fine coffee filter, so to speak. And then we retained pretty much all the stuff that was in the water on that coffee filter, and then later on rinsed everything away that wasn't the DNA. And then we kept the DNA from whatever was swimming through that water, and that's going to be part of our analysis as well. So we will be able to not only see what was on or in the sediment, but also what was swimming around those depths.
And Andrew, how much water can each Niskin bottle hold?
So those particular Niskin bottles will hold 5 liters, I believe. I could be wrong. It could be 2.5. I think they're 5-liter ones. So we can take. Well, I mean, we can put as many Niskin bottles onto the vehicle as we want, but really, we use the CTD carousel for large volumes. It's more for sort of opportunistic. If we do some sampling on the seabed and we want to get the water right beside it or right above it, then having them on the ROV is a much easier way. If we're doing sort of full sampling through the water column from bottom up to the top, then the CTD and the rosette, the Niskin rosette is a much more efficient way, and we can collect 10 liters at a time in each of those, and we've 24 bottles. So that's a lot of water that we can, 240 liters of water that we can collect in a single cast with that system.
Yeah, usually we do use the Niskins on the subs and RVs for particular things that we see during the dive, like if we see something unknown or for example patchy seafloor or species of coral that we've never seen, or have a dive that's so spectacular that we want to have water samples taken of it, then we would use the little bit smaller Niskins on the RV, whereas the full vertical profile of the water column, we use the CTD rosette for that, which holds a lot more water, as Andrew just said.
And just FYI, a couple of the team kept some of that water from 4,600 meters as keepsake. So a few of us have like these little vials of seawater from 4,600 meters.
Very cool.
Yeah, it's all about the personalized souvenirs that you take for yourself from like everything's exciting, but that one was particularly exciting, and then you want to keep a tiny little memory for that.
What was it you particularly liked about the sediment down here, Larissa? I remember you were very, very excited about it.
I mean, I am usually very excited about sediment. But just when it got to the surface, it still was very cold. I remember that I think the temperature reading at that depth was like 4, 5, 6 degrees. So it was like pretty cold. And the sediment was still really, really cold when it got up to the surface, which is unusual because by the time usually the RV comes up, and like in the areas we've operated and the depth, the depth we've operated so far, the sediment was never that cold, or like any sediment that I've worked with. And it just had such a beautiful pale color compared to all the mud that I've seen before. And I
Wait,
think
we
it
have
was
to
just,
talk about this fish. What are we looking at here?
It
is it
it looks
this
like
no,
a
snailfish? No.
snailfish or a conger
Starfish?
eel. I remember we were really annoyed at this point because we couldn't quite focus on this fish.
Yeah, there's also a lot of stuff flying
In the
around in the
water
water
column, yeah.
column.
Yeah, that's the issue with sort of sampling, sort of sitting down on these sort of very silty clay bottoms. Is you have to sit down to sample, and as soon as you sample, then you come back up again, and it sort of sticks to the ROV like it was sticking to that our little scoop. So yeah, you spend forever then just dealing with sediment as you fly around, unless you keep moving.
It
Yeah, I
looks
mean
a bit
we're
like a
deep at this point, so how common is it to like how many species of fish live at these sort of depths?
You get fish that are specially adapted for these deep depths. Cusk eels, conger eels, different types of snailfish. Snailfish being one of the deepest fishes that we know of, being found more than 8,000 meters. So I think, Larissa, I'm not sure. I think this might be either like a snailfish or a cusk eel. We can't really
I
tell.
I'd go with
What do you reckon?
snailfish.
Yeah?
I'd go with snailfish just by the grumpy look on its face. I feel like that's just snailfish always like they always just look like they're not having a good day.
It's got that
Um,
foamy face.
yeah, I like the little fins. Yeah, I'd probably go with snailfish, but like you never know. They're actually super interesting. I recently read that some snailfish are used in molecular biology, which is what I do, because they do have some special type of genes, um, or like they have mutations in genes that do now create proteins that help them survive in the deep. So they had like undergone a change from like smelling to tasting, and like different adaptations. Um, and these are of particular interest to like figure out how they actually do survive there. And then once we figure that out, we might also know why they look sad all the time.
What I love about them, there's this really interesting fact that they like to latch onto things as well. So they sometimes hop on a crab and lay their eggs in the gills, which is not pleasant for the crab, I don't imagine. But yeah, so that's really a really interesting little fish.
Yeah, they do look like really big tadpoles somehow,
Yeah.
like tadpole fishies thing. Is that another sea cucumber there?
Yeah.
Yeah, it looks like it. Do you remember how much seagrass we found at this depth as well?
Yeah, that was crazy. Like all the little circles where they would like get entangled in themselves. Um, I think it also was like mainly one type that like really long bladed. I forgot the name, but there were so many of those like that. Probably is like one of the best visual representations of like carbon transfer to the deep, just like all the seagrass literally rolling down the hills and
Yeah.
then just accumulate at 4,600 meters.
So is this the same species as the gummy squirrels, or is this a different kind of sea cucumber that we're looking at?
It looks the same to me.
Yeah,
Except it's
certainly
on the
some.
move.
Yeah.
Yeah,
Is it using that sail to swim or is it just kind of floating?
it looks like
So
It
it's
looks like
kind
it's
of
just like
floating, kind of
floating a little, right?
steering. Yeah,
Yeah.
What do you call that movement? Hopping? I don't know. It is definitely like it looks a lot more bloated, but you can see that it's definitely a sea cucumber, and it definitely looks the same as the ones we were seeing previously.
Yeah, the grape flavored, gummy
A great
squirrel.
flavor.
Exactly. Sheena, what is that sort of white circle? That
The
looks, is that
eye.
an eye, or do they have eyes?
I think that looks like the anus. So it looks like where all the poop comes out. And I remember seeing lots of like, I don't know if you remember this. We saw lots of like squiggly, sandy poops around that we thought was definitely from these sea cucumbers. They were very close to the sea cucumbers as well. And they were like spirally, which we thought was really interesting. Did we take a sample of any of those, Larissa? Can you remember?
I can definitely remember seeing them, but I don't think we took a particular sample of those. Not that I can
No,
recall.
I don't think so. We need to go back, Sheena.
Yes, I agree.
Go back.
I already have
Gotta get
ideas.
those poop samples.
Yeah, I feel like we're not running out of ideas here.
I think what was also really nice to see as we, came shallower, was there was lots of muddy patches, but then we had these rocky outcrops, which sometimes had life living on it, like lots of different types of sponges, and some coral, which was really interesting.
Sheena, Larissa, could you explain like what is the difference between a shallow water coral versus a deep water coral? With shallow water corals mostly relying on the sun for photosynthesis, but there's no sun down here. So what are these deep sea corals kind of feeding on?
Do you want to start, Larissa?
So the shallow water corals, as you said, like do rely on sunlight, most of them. So they have like those tiny little algae sitting in their tissue, and they just made a pact on the algae using sunlight to produce energy and share that energy with the coral. And in return, the coral supplies them with nutrients and shelter. And you're completely correct that the second we run out of sunlight, that whole pact is no longer possible. And the deep sea corals had to like come up with a different mechanism to do that. Um, Sheena, what you want to talk about the deep ones?
Cool. Yeah, the deep guys. They're so interesting. You get so many different types of deep sea corals as well. But they kind of have little tentacles, and they collect the snow that comes out from the surface and rains down on them because they don't move around really. So they had to come up with a different way of feeding. So they don't rely on the sun at all, and they rely on sea snow and whatever's in the water column.
What is sea snow, Sheena?
Sea snow, it's different bits of dead things coming down from the surface all the way down to the bottom of the sea. So if you look in our video, you can see lots of specks in the water. And we call it sea snow because it looks exactly like that. It feels like it's snowing underwater. And essentially it is, but it's snowing detritus and parts of dead animals and bacteria and all sorts of little clouds of goodness or yumminess for the corals.
Oh, look, these
Isn't
are
this
the
isn't
seagrass
this some of
babies.
the sea? Yeah, the seagrass.
Yeah,
There's lots of patches. This is where we were seeing them, isn't it?
yeah, it's so
Yeah.
crazy that they just like assemble and then just get like spun in circles and form those like little patches that almost look like they would grow there, but they surely don't. They just get transported down there and then just aggregate in those little pillows that I assume probably also provide a really, really nice buffet for everyone that's like
Mm.
down there out and about looking for food.
And I think what was really cool is we saw seagrass at every depth we went to, especially at the deeper depths. in all of our dives. And yeah, we were really, I guess, not surprised, but we were like, whoa, that's crazy that there's so much seagrass down here as well.
Yeah,
And
especially the
on
amount.
the
Like it
ridge,
was
so
just
on the
so
east
much.
side, there would be lots of the small islands, coralline islands, islands like Poivre and Daros, and they have a lot of seagrass beds around these atolls, and it's like a clear transfer from the surface all the way to the deep depths.
Are the seagrass beds, they're not all the same species, right? Like,
No, we get like 7 different types
like
of
within the,
species here,
yeah.
but the ones that we were seeing, the species that we were seeing mostly at the in most of our dives were the long-leafed, the broad-leafed
Yeah, those like
seagrass.
really long, almost blade-looking things compared to
Yes.
like the smaller, broader ones that we also saw, like in the coastal areas.
Hmm.
I just want to take a moment to recognize Andrew and that very efficient 1-2 punch on the Niskin bottles there. Not even a hesitation. So well done.
You blinked, you would have missed that one. That was, yes.
It was,
Every once in a while, you hit it first time.
I think I could not
Wasn't
do
that
that,
me
like my
doing that, Andrew?
three-dimensional vision.
Yes, yes.
Didn't you give me the controls? That's why it was so smooth.
Yes, that was Sheena. Yes. Sheena was flying down here. I remember. Pretty sure you were in the pilot seat at one point.
I wasn't allowed to touch the controls, which is fair enough because I'm very clumsy. We wouldn't have had an ROV otherwise.
But the RV pilot chairs are the most comfy seats we have on the boat, though.
Yeah, they're pretty comfortable.
I
I
don't know
will
how
give
they
you that.
don't fall asleep.
Because it's
In
so
that bag,
exciting, yeah.
so in that bag, we did we brought the styrofoam heads with us. I was one, wasn't it? One big giant one, and everybody who was on the mission signed it. So it was like this size when it went
Yes.
down, and obviously if you put it under pressure, under 4,600 meters of pressure, the whole thing crushes down in perfect proportions down to about this size. But you can still read all the names. You can still see all the pictures that people have made on it. It's just tiny and in miniature. It's a very cool little thing to do. It's sort of to show kids and other people like what pressure actually does and how crushing the pressure is down there. So a nice little keepsake. Where is it now? It's in the Seychelles, isn't it, Sheena? Somewhere?
Yes,
Probably
it's
on your
currently
desk,
sitting
I would
with me,
imagine.
but we're
Yeah,
we're
I thought so.
donating it to the museum later this month. So
Oh,
we're
cool.
gonna have a little
Cool.
ceremony and everything, and it's gonna be quite a big deal. And we're gonna hand it over to the museum as part of our history.
Did you do a keep a large one which wasn't crushed, or are you gonna have to get the band back together to recreate the big one, and then you can give them both?
We're gonna have to recreate the big one. Unfortunately, we've got lots of photos and videos, but yeah.
Yeah.
I think that was one of the highlights of the dive as well. Everyone was really interested to find out what happened to this head and what shape it would be in once it came back up to the surface.
Looks like we're about to take more sediment. I wonder what person requested that.
Yes. I think we're coming towards the end of the dive. We did samples at the start. I think we took the last 2 water samples, a few more sediment samples, and if I recall, it was around the sort of depth that we started to wrap things up. You see down in the corner of the screen. Yeah. So we have on the screen here, on all the overlay for Sheena and the other scientists sort of later on, we have along the top is the actual heading that the ROV is on, and obviously on the right is the depth. The altitude is below that. But down below, is the lat and the long, which is blanked out. And then on the left-hand side, so the dive number, the mission number, the time, and the date. So at any given time, you can then have a visual reference straight away to understand what date this happened, where it happened, what the dive number was, to be able to then go back to our electronic logs and sort of look up all of the things that were said and done during the dive. And it's all, so all the metadata from all of the dives is captured in such a way that it's easy to recall again. Because, I think we ended up with over 50 dives in the 5 weeks we were there, or something crazy like that. We were diving every day, and some days twice a day. So there's quite a lot of hours of footage for Sheena to get through before we go back again.
I was very greedy with the diving.
Yeah.
But you're only there once, so you gotta get as much as we
That's
can.
right. Yeah. I think we all agree we got to go back. We got to go back to the Seychelles. Such a beautiful, stunning country, and the diversity across that. What is it, from one end to the other? It's nearly 1,000 nautical miles. One of the biggest EEZs in the world, isn't it?
Yep. It's massive. I mean, 99% of our country is water. And 96% of our whole country is deep water, and we've investigated this much of it. So there's so much more to do.
There's so much more sediment, so much more water, so much more gummy squirrels to see. Maybe we'll find other flavors.
And Andrew, I'm very curious. What is that pad with all the different colored squares on it in the tray?
So when we do sort of our high end media content, we bring a color chart with us so that we can then white balance and black balance the camera. So the footage that we collect is sort of super high, 4 to 8K quality. But we have an expert here with us right in the middle. Eben is our resident media expert. But yeah, it's very important that those cameras give the best images back. So the content that these guys create is sort of awe-inspiring and stunning to look at.
Yeah, it's a tool for getting your color correction just right, so you can compare a red to a red down at depth there. So then when you can match them up and make sure that your color is spot on.
Yeah, on these sort of dives, because we're very much sort of the Swiss Army knife, and we have some good cameras on here, but they're not they're not uber good. They're not like our Benthic 4 and 8K cameras but because we just wouldn't have room for that and be able to do all of the sampling. So that's why our sort of footage is sort of that. It's a lower quality, but it's good enough for the scientists to see and to be able to do their analysis from. But it's not what you're looking at when we create that amazing media content. That's a whole different rig of cameras that we can put on here, or the subs have on them. So yeah, cameras for courses, as they say. Oh, we must be coming up soon. I think I feel like I've done the whole dive again.
I was reading up about the Amiran's Trench, actually, and they've got so many theories about how it came about, from it being a subduction zone to an asteroid, to the asteroid's been disproved, to even it being formed when Seychelles kind of broke away from India. So it's really interesting geology, and they're still unsure about how the Amiran's Trench came about, because it doesn't receive as much attention as, the Marianas Trench or a lot of the other trenches in the world. So there's definitely a lot more work to do, not just the biology, but also the geology of this area.
Yeah, we literally scratched the surface, or scratched the bottom, I think is probably more correct.
Scratch the bottom, I like that. What was your favorite
Yeah, I
part of
think
this
we're on our way
dive,
up now.
Andrew?
Uh, I think making it to the bottom and back up. Well, actually, my favorite part of the dive is coming up in a minute. I don't want to spoil it for anybody, but this was definitely what's going to happen pretty shortly. Was pretty epic.
Yeah,
And
so definitely on our way up now. 960
yeah,
meters.
yeah,
That was the best ascent ever.
yeah. I like them that quick. No, no, but like making it to the bottom is pretty cool feat in one go and back up again, and then like you said, we're exploring all of the time, and you never know what you're going to come across, and it could be two after 2 meters on the way down, it could be like 6,000 meters at the bottom of the ocean, or it could be just you're sort of wrapping things up, getting ready for lunch, and you just things happen. And that's the beauty. That's the beauty of what we do. It is exploration. It's ocean exploration, and yeah. We were coming back up. We were, like I said, 900 meters. Sort of all thinking about what's coming up, and then all of a sudden we had a sort of a friend come and see us, which should be along any minute now. Yeah, an impromptu visit. Yeah. And
Yeah, thinking about sample processing and then just like, oh God, we're actually not done with the exploration and the seeing cool things.
and a lot of it, a lot of these things that happen, it's sometimes we even miss them. Oh,
Yeah,
it's like,
there we go.
There it is.
So cool.
So cool. Oh my gosh.
I never get bored of seeing that, like just over and
No.
over. And it was almost missed the first time. It was like somebody said, "Whoa, what was that?" But because the cameras, you never stop running the cameras, like they're always running, and then they come back and it comes back again. Okay, and so we're looking at one camera. We've got 4 other cameras that capture that interaction. Sort of the one below, the one above, and one of the side ones catch it as well. So that's why you always.
Yeah,
Here we go.
here's
the
another
lower
angle.
one. Yeah.
So beautiful.
So
Very
just,
cool.
So beautiful.
I actually looked up the size of the photophores because it just was so bright, just like literally flashing. And the photophores are the size of lemons,
Yes.
like
Huge.
Amazing.
like that big. That's just so crazy.
So what are we looking at? What species?
It's a Dana octopus squid.
Or Teningia. That's the major group it's in, and
They're like,
it's
I
so
think they're
cool
one of the
because
biggest. Wait, that's
they're one of the they are one of the biggest, and they, unlike other squid where they have the 2 like tentacles, they have the arms and those like feeding tentacles. These guys don't, and you have these photophores at the end of like 2 of the arms, and it's like if you think about it, it's like an eye, except that it doesn't absorb light, it like exudes light, which is really, really cool.
yeah, it's just
And there's
so
like
crazy.
a lot of theories as to
Just
why
like
they use
something
it,
out of
whether
nowhere.
it's like to scare things away or to stun their prey. Yeah, so there's a lot of theories around why they use the photophores.
Yeah, probably like territorial behavior or like mating purposes, something. Could be so many things. I feel like when you live in the dark,
Communication.
light is the one thing that you need for everything. To like get away with your life, find food, find a partner, get away. Just find your way around. Like you just need it. You gotta be your own. Bring your own torch.
All right.
Little
There's
fishy
Ocean
on the way back up.
Explorer.
Bottom of the boat.
Back home.
Okay, hauling out.
That's always my favorite bit, when we actually get it back on board again. That's quite nice. It's like, hey.
Oh, cool. Well, that's it.
All right.
Wow, very, very cool, guys. Well, boy, yeah, just like that, we've come to the end of our record-breaking dive. So I want to thank my panel of distinguished colleagues for joining us from all over the world. And of course, thank you for watching. We'd love to know what you think of this new format that we're playing around with. So let us know in the comments what you thought or what you'd like to see next. So until next time, thanks everybody. Really appreciate it.
Bye.
Thanks, Evan.
Bye.
Bye.